Bismillahirrahmanirrahim. Let's begin. Uh, so the following is papers, the first variant 6-1 of ATP from October, November 2020. We are going to do a run through of this paper, which basically means that we are going to see how the paper works. Now, what I advise you to do is, once again, as I said, I am doing this run through. I will make sure that I explain everything in detail. For those of you who just come in, I just want you to turn your mics off and listen to me very carefully, okay? We are going to do a run through of this paper. I want you to have a paper and pencil ready and take note of some important things. Although the majority of the run through is going to be done by myself. This is the latest. This is the latest paper. Okay, and we are going to look at the consultant marking schemes and the examiner reports. Okay, so right away, let's begin with our paper. So it says, and you're going to see I'm going to do a lot of scribbling on top of this page. So it's very important that you understand. Okay. Okay, can you please turn your mics off, everybody, and listen carefully. I don't want any distractions. When tissue from some plants is cut, I think I should paste this paper on my OneNote so that all of the scribbling that I do is worth it. Okay. Insert files. Okay. Papers. Sorry for the sound effects, but I can't help it. Okay, so it's this question paper that we want. Okay. Okay. So when the tissue from some plants is cut, the cut surface turns brown. Okay, so as you guys know already, I've told you many times that we need to be very critical with all of the information that has been given in the question. So they're talking about a tissue of some plant that is cut and the cut surface turns brown. Okay, it turns brown. So it appears that he's trying to talk about some color change. Okay, and right away we know that the color change maybe has to do with some of the food tests. Okay, but then it says this is because enzymes are released which cause reactions between chemicals in plants and oxygen to produce brown substance. Okay, so I'm going to rewrite this for myself. Some chemicals, they are reacting with oxygen to produce brown substances. Of course, all of this is being caused by enzyme, because this is what he's saying. This is because enzymes are released which cause reactions. So enzymes are responsible for some chemicals in the plants that react with oxygen to produce brown substances. Now, all what I did was, and take this as an advice, that whenever you are reading the lengthy text of the ATP, it is very important that you change that into a visual form. You rewrite it in this way. It really helps you to retain the information. The photograph below shows two slices cut from a banana. Slice A was exposed to air for 2 hours. Okay, so this was exposed to air for 2 hours. Okay, all what I'm doing is, once again, you can clearly see I am restating excuse me, I'm restating the information just for the sake of my clarity. Okay, so it's exposed to 2 hours, and slice B was freshly cut. So this is not exposed, this is just freshly cut. And as you can see from the diagram, the B cut seems fresher as compared to the A. I mean, the way आप देखते हैं केले को अगर आप ज़्यादा देर रख दें, तो that's basically how bad it goes. Okay, the freshly cut slice was creamy yellow in color. Okay, so color here is creamy yellow. Have they talked about the color of the A? Well, they've given us a hint that some brown substance is produced. Okay, so whatever the case is, let's move forward. Now, they're saying some students decided to investigate the effect of different treatments on the rate at which banana turns brown. Now, it is very important, bachcho, that whenever you are looking at an investigation design, it's important that you see what is being investigated against what. So if you look at this statement, आप देख रहे हैं कि I'm spending a lot of time on the statement of the question that is being asked. Okay, so some students decided to investigate the effect of different treatments on the rate at which banana turns brown. Okay, so they are basically applying something. They're giving different treatments, and they're seeing the rate at which. So the rate we know has to do something with the time factor. Okay, again, once again, reiterating the fact that the rate has to do with the time factor. Okay, all right, then it's further says, please listen carefully, this is important. They cut 3 slices from the same banana, each 1 centimeter wide. Now in experiments, you will come across ideas which in which they will make sure that there are certain things that are controlled or standardized. So for example, they're taking 3 slices of the same banana, each 1 centimeter wide. Now they could have easily done the other way around. For example, if this is representing 1 centimeter wide, okay, they could have easily taken some other pieces like 2 centimeter, but they didn't do this. Okay, they only took 3 slices that were 1 centimeter each. Now the question is, why did they do this? You have to understand, in ATP paper, when you're reading through the text, you have to be very critical. Ask questions. Why is this happening? So remember that whenever in an investigation, the size of something is being controlled, yes, some temperature is being controlled, yes, some volume is being controlled, and by control I mean that it's kept the same for example, for all 3 purposes. We say that this is basically controlling the variables. I'm going to pause here and I'm going to talk something about generally about investigation design. Let me use this side of the board. Once again, when you are talking about investigation design, you need to understand that at the end of the day, if I want to explain you in a very simple word, what is a biological investigation? Yeah, chale biological na bhi ho, let it be any scientific investigation. You are basically seeing effect of one thing on the other. Okay? On the other. ठीक है, मैं सिर्फ समझाने के लिए इसको verses कर देता होता हूँ. I mean, the one thing, you don't call that thing as thing in the investigations, you call that thing as variable. So you're seeing the effect of one variable on the other, and you call both of them as, the first one is going to be called as the independent variable, okay? And the other is going to be called independent variable. I'm sorry. Oh, एक independent है और दूसरा dependent है. My bad. I'm sorry. The other is dependent.
Sorry, one note to बड़ा अच्छा.
Okay. So independent variable and independent variable. Okay. Up, we have to understand that what is the difference between dependent variable and independent variable. देखिए, हमने पहले भी ये बात की हुई है. The independent variable is something that I, as a investigator, I myself am varying. Okay. यानी कि, for example, let me give you a very simple example. For example, if you're trying to see the effect of temperature on enzyme action, I'm just giving you a random example. Okay, this is not in the context of this question, but it's generally explaining. So what I do is I check the enzyme activity or action on 5 different temperatures, for example, 10 degrees Celsius, 20 degrees Celsius, 30 degrees Celsius, 40 degrees Celsius, and 50 degrees Celsius. Okay? These are 5 temperatures which I deliberately change, okay, and I check the activity of enzymes based on this temperature. Now, you need to understand that changing the temperature is in my own hand as an investigator. I myself am varying that. Therefore, we call the temperature in this example as the independent variable. Okay. And as a result of varying the temperature, yani ki for example, if I'm transitioning from 10 to 20, for example, what that means is that because of increasing temperature, the enzyme action, for example, the rate of enzyme activity increases. Okay, so this thing is dependent upon myself changing the temperature. And now, how do I know that the enzyme action is increasing? Maybe a gas is being produced, for example. A gas is being produced. So how do I know that the rate of reaction is increasing? Maybe we are using the counting the number of bubbles, for example. Okay, to count. So for example, if on 10 degrees Celsius, you have 20 bubbles being produced, then on when you increase it to 20, maybe 40 bubbles are being produced. Okay, so the dependent variable, but you remember, is something that you directly measure. Remember this with the D. Okay, so directly measure or independent variable है जो I myself, remember, I'm trying to make things simpler. Okay, so I vary is the independent, and the one that I directly measure is the dependent variable. Now, if I was to say that the enzyme action is the dependent variable, this would have been incorrect. Let me repeat, listen to me carefully. If I say that the enzyme action is the dependent variable, then this is incorrect. And the reason is because I am directly measuring the number of bubbles, which no doubt is representing the enzyme action. But in technicality, I would ensure that I include the thing that I'm measuring directly as the dependent variable. Okay, so this is basically the dynamics of what an investigation is. Acha, iske alawa now apart from temperature and enzyme action, maybe there will be some other variables that need to be controlled. For example, if we are taking the volume of solution, if we're talking about the concentration of enzymes, for example, फिर इसी तरह, I mean, of course, pressure would in no way affect this experiment. I mean, you need to think according to the context of the experiment. What are other variables that need to be controlled in order to make sure that it's only temperature that is affecting the dependent variable and not the others. Okay? So coming back now to the question, यहाँ पे आप देखें कि उसने जो ये कहा है they got 3 slices from the same banana. The fact that they're taking it from the same banana and the fact that all 3 of them are 1 centimeter wide, both of them happen to be the controlled variables. Okay? So again, that's very important. Now let's proceed. One slice C was placed in a beaker of HCl for 2 minutes. It was then removed and placed on a white tile. Now, as I said, I'm reiterating again and again. इतना ज़्यादा टेक्स्ट है, अभी आप सोच रहे होंगे कि मैंने इतना टाइम इसी पे लगा दिया. No, यकीन करें, you spending time on the text is worth it. So, I'm going to make a visual for myself. One slice C was placed in a beaker of dilute HCl for 2 minutes. So I'm going to draw a beaker for myself here. यहाँ पे draw करते हैं. ठीक है? One slice C was, okay. So the beaker is filled with dilute HCl. It has HCl, which is dilute. Okay, I'm labeling this for myself. And then one slice C was placed in this. Okay. For 2 minutes. For how many minutes? 2 minutes. Okay. It was then removed and placed on white tile. ठीक है, उसके बाद उसको remove करके किस पे रख दिया जी? White tile पे रख दिया. देखेंगे मैं सारा कुछ draw करा. Now this drawing all this is not a requirement from the Cambridge, but you will see how doing this really helps you to understand how things work. Okay, we're done with this. One slice D was placed on a white tile and cut into many pieces. Okay. So you have another स्लाइस टाइल उस पे बहुत सारे पीसेस कर दिए इस एक बनाना के one slice, sorry, the slice is cut into many small pieces. Okay, so these are many small pieces and they're labeling this as D. हम इसको D label कर देते हैं, many slices of D. ठीक है? And then third slice E was left untreated on a white tile. ठीक है? कोई acid नहीं डाला, कोई pieces नहीं किए, it was left untreated. Okay, so the students observed the slices and recorded the color of each after 5, 10, and 20 minutes. Okay, now if you think about it, what is that the students are recording and measuring? I mean, yeah, आप यहाँ पे पूछ नहीं रहा, लेकिन क्योंकि मैंने आपको ऊपर इन्वेस्टिगेशन डिजाइन का आइडिया दिया है थोड़ा, तो आप क्लियरली देख रहे हैं that the stuff that is dependent variable that's being directly measured. So maybe in this investigation, and I'm saying maybe because we don't know as yet, they are saying the students recorded. Now we know that the thing that is recorded can be the direct. Sorry, not the direct. So, my bad. It's the dependent variable. Just give me a second. It's the dependent variable. Okay. After 5, अच्छा आप देखें कि टाइम ड्यूरेशन क्या है. पहले 5 मिनट्स के बाद चेक किया, फिर 10 मिनट्स के बाद किया. 5 और 10 मिनट्स में 5 मिनट्स का गैप है. So this gap is even, it's 5 minutes, and then they increase this gap. 10 to 20 minutes has a 10-minute gap. Okay, so the time duration with which they're observing is actually increasing. पहले 5 minutes के बाद, फिर 10 minutes के बाद, and so on and so forth. After 5 minutes, they also measured and recorded the pH. Okay, so not only they're recording the color, but they're also recording the pH. So I have two things that can become the dependent variable. Why the dependent variable? Because that is what they're directly measuring. Of the cut surface of each slice, the results are shown below. Okay, now let's look at the results and let's try and understand what they've written in the given in the box. Okay. After 5 minutes, HCl treatment. अच्छा, HCl treatment pH 2. जाहिर बात है HCl की जो है pH है वो 2 है. Okay, although it was dilute, but the pH is 2. Creamy yellow color है, cut up slice है, और pH, अच्छा cut up slice की जो है pH है वो 6 है. ठीक है. Did they apply pH acid on the cut up slice in D? Here, no, they didn't. Okay. उसका जो कलर है वो पेल येलो ब्राउन है, और untreated की pH 6 है और creamy येलो है. Now what I'm going to do is, again, देखिए इतनी इंफॉर्मेशन है, अगर आप उसको अपने लिए साइड पे नहीं लिखेंगे आपको क्लैरिटी नहीं आएगी. I want you to pick and choose these two things from the table, the pH and the color. So the pH, so what I'm going to do is, उसने देखो यहाँ पे टेबल भी बनाया है, लेकिन नीचे हम करते हैं कि after 5 minutes जो है आपका जो pH है वो 2 है, और जो आपका कलर है बच्चों वो क्या है? The color is creamy yellow. Creamy yellow. ठीक है? Okay. For after 10 minutes, let's look at the pH. What's the pH? The pH is, have they given the pH? Well, अच्छा नहीं, I'm sorry. 5 minutes पहले हम 5 minutes पे तीनों को देख लेते हैं, फिर हम आगे आते हैं. So 5 minutes पे जो आपका पहला था piece था, this is talking about C. We're talking about C after 5 minutes. Then you are talking about D, which is the cut up slice, which is pH 6, and the color is pale yellow, pale brown. ठीक है. और जो E है ये वाला है, ये वाला जो E है, इसका क्या चक्कर है? E पे the color is creamy yellow और pH क्या है? 6 है. इसकी भी pH 6 है और इसका कलर भी creamy yellow. ठीक है. And after 10 minutes and after 20 minutes they've done the same. HCl treatment. अच्छा, फिर after 10 minutes पे उसने pH नहीं लिखी, उसने HCl treatment लिखा हुआ है, कलर लिखा हुआ है, सिर्फ कलर लिखा हुआ है तीनों का. C का उसने क्या लिखा हुआ है? कलर लिखा हुआ है. कट अप स्लाइस जो D था उसका भी कलर लिखा हुआ है. And then the untreated slice of that was E, that is also the color is being mentioned. Now After 20 minutes, again, HCl treatment, creamy yellow. They've only given color for C, cut up slice. They've only given color for D, and they've given color. Now, अब आप देखें कि we can clearly see that the student has actually made a mistake. And that mistake is that, अच्छा नहीं, उसने यहाँ पे लिखा हुआ है, after 5 minutes, they also measured and recorded the pH of each slice. Okay, I mean, he's only measured the pH after 5 minutes. After 10 or 20 minutes, उसने pH को lift नहीं करवाई, उसने सिर्फ color देखा. Okay, now it says enter the student's data in the tables. Now you need to understand that excuse me, ये जो आपका skill set है, this is a table. You are going to translate or shift this information from the text that the student has written into the table. Okay, and this is a skill that is basically being tested. So now let's try and make sense of the table first. Okay, I mean, imagine if you were the student and you were asked to make a table of all of these observations, then this is basically how this would have been. Extreme left of the table. By the way, there are instances where you may, come across um, time frames. And वैसे O levels में table बनवाने के लिए वो कहते नहीं, लेकिन you have to understand that the extreme left side of the table is always independent variable. So you will always find usually time, that's in minutes. And then for color, उसने देखे C, D और E तीनों का लिख के दिया और उसने pH का अलग टेबल बनाया हुआ है. वो चाहता तो pH का साथ भी कर देता, but again, and why are we considering color and pH? Because that's what he says in the question. That's what he says in the question. Look, he says they recorded the color and they recorded the pH. Okay, so let's look at the table one by one. So after 5 minutes, जो color था slice dropped in HCl, C में वो क्या था? हम आते हैं ऊपर अपने उसके उनका जो उन्होंने लिखा हुआ है. So you can clearly see that. Okay, so we have both of the things now. We can see that after 5 minutes, the color was creamy yellow. Okay, for C. Okay, so यहाँ पे हम क्या लिखेंगे? Creamy yellow. Okay, and then for D, slice that was cut in small pieces. ये देखिए slice कटा, pH लिखी हुई है और आगे उसका color दिया हुआ है very pale brown. Okay, so we write it as it is, very excuse me, pale brown. Okay, and then you have E, the untreated slice. ये देखो यहाँ पे लिखा हुआ है after 5 minutes की बात हो रही है, उसका color क्या है बच्चों? Creamy yellow. So that is creamy yellow. Okay, that's for after 5 minutes. Now, after 10 minutes, we have to now tell what was the situation. So for C, again, we've seen the color is creamy yellow. We've already done that. देखिए कितना फायदा होता है that we scribble on the text. It really does the work easy for us. And then for D, after 10 minutes, so we're talking after 10 minutes, or D पे हमने ये देखिए color पहले हमने mark उसको किया है D, that is pale brown. And then for 10 untreated slice is very pale brown. Very pale brown. अच्छा यहाँ पे very pale brown ही लिखना है. अगर किसी ने खाली brown लिख दिया तो उसको mark नहीं मिलेगा, because that's the name of the color that they've chosen. And for 20, you have for C, देखिए after 20, C पे सबसे पहले creamy yellow है. Again, that's creamy yellow. And for D, it is brown. And for D, it is pale brown. And now let's talk about pH. Size, अच्छा, अब वो pH की बात कर रहा है. C पे उसने pH जो है वो D, अच्छा, he is talking about pH, but then he is not talking about the time frame. So since उसने यहाँ पे खुद कह दिया है कि after 5 minutes they also measured and recorded the pH. So it's the pH they only recorded after 5 minutes. That's why you only have one table here. So pH देखें क्या थी, पहले 2 थी, फिर 6 थी और फिर दोबारा 6 थी. ठीक है, so it is 2, it is 6, and it is 6. So if you look at the distribution of marks in the exam, you will basically get 1 mark for this correct and the remaining 2 marks for all of this. ठीक है, अब हम एक काम करते हैं,